Okay, so here we have our enzyme lab. So what we're looking at today is we are, again, testing the hydrogen peroxide. We've got our raw liver. We've got our 2 test tubes and a little stick to help us push the liver in if needed. Remember, the hydrogen peroxide is poisonous. You would never want to eat or drink anything in the lab. So what you were asked to do today was you were going to take the hydrogen peroxide and you were supposed to measure out 2 milliliters of it. So you poured it into the graduated cylinder. You put it up at eye level and you measured the exactly 2 milliliters, and you went ahead and poured it into your test tube. Now remember, your body, as it said in the lab, actually makes hydrogen peroxide, which is poisonous and toxic to your body. Your cells do that. They make hydrogen peroxide during the process of metabolism. So your body is making a poison. Fortunately, inside your body you have a liver, and that liver has an enzyme called Catalyse, which can help break apart that poison so it does not kill and hurt your cells. So the next step is to go ahead and take a piece of raw liver, and it asks us to go ahead and set that into our test tube. And if it sticks, we can go ahead and push it all the way down, as so. And when we do that, you can see that the liver is reacting with the hydrogen peroxide. It's bubbling, and we're creating a gas, so we definitely have a chemical reaction that's going on here. And it's actually the enzymes in the liver, specifically the enzyme Catalyse, that's breaking apart that hydrogen peroxide. So you can look at the chemical formula and see what that liver is breaking it apart into, but it's making 2 substances, one of which we can see is a gas. And now, this wasn't part of the lab, but we can go ahead and test that gas because we've tested the gases before, and we can do what's called the glowing splint test. And so if we take a wooden splint and we light it on fire, we can then go ahead and do the glowing splint test on this gas, blow it out, and if we stick it into the gas, what we'll see is that it reignites. That's because the gas that we've created is actually oxygen gas. Okay? What we want to, though, figure out is what is going to happen next. So one of the things that you were supposed to do is feel the bottom of this while the reaction was happening, and if you do, you can actually feel it is getting warm. So one of the questions it's asking you is this an endothermic or exothermic reaction, and you can look up those vocabulary words if you don't know already to see, since this is getting warm, that tells us that this is a certain type of reaction, and so you can classify that and answer that on your sheet. Okay? The next thing we want to look at is this enzyme reusable? And so we want to do 2 tests with that. The first is once this is done reacting, we're going to test this liquid. So what I did is I went ahead, since you can still see we're still getting some bubbles here. It's mostly done, but I don't want to wait for it. I went ahead and ran this one a few minutes ago, about 5 minutes ago, and you can see that we're not creating any new bubbles off of this piece of liver. So this has completely reacted. So it's asking you to do first to take just the liquid and pour the liquid off into a new test tube. So I'm just going to pour the liquid off, and I'm going to save the liver, the used piece of liver, and here's the liquid that I used. Okay? The next question is if I were to add a fresh piece of liver to this liquid, what would happen? So you need to go ahead and pause the video and make your prediction. What would happen, and specifically you want to be able to compare what would the reaction rate be. So if I added a fresh piece of liver and we considered that reaction before was a 4, when we saw these bubbles getting created, that speed of that was a 4, how if I put a fresh piece of liver into this liquid that I just poured off, would the rate compare? Is it going to be the same? Would it also be, happen just as fast? Would it also be a 4? Would it happen even faster with this liquid that's already been used? Would it happen slower? What would you get? So make your prediction, write it down, and now we will test it. So pause the video if you need to. Okay, here's the fresh piece of liver. We're going to push it down, and what we see, of course, is what happens? Well, there's still the old bubbles at the surface, but you can see we're not seeing any new bubbles created. That's because there is no reaction happening in this at all. Okay, so you can think about why that is. If you look at what you're creating when you're doing this reaction and what are the two substances that hydrogen peroxide breaks down into, it should make sense to you why there is no reaction when we put the piece of liver into this used liquid. It's because the hydrogen peroxide in this liquid has already been completely broken apart, and so this liquid does not contain any hydrogen peroxide, and that's why we don't have any reaction here. Okay? And once you figure out what the liquid actually is, it makes even more sense why the liver is not going to react, because otherwise your body would be reacting all the time. Now, the next thing that you want to look at is, here's the piece of used hydrogen peroxide. So you saw when we looked at the used liquid, nothing happened when we put a fresh piece of hydrogen peroxide in here. Now we want to figure out what would happen if I were to put fresh hydrogen peroxide into this used piece of liver. Okay, so we had, this was a fresh piece of liver into the used liquid and nothing happened. Now I want to know, is the enzyme reusable? So I'll measure out another 2 milliliters, and we want to see, again, hold it up at eye level, make sure it's exact. In this one, we used up all of the hydrogen peroxide at the reactants, and so no reaction happened when we put the liver in there. And you should make your prediction. This is the next question. If we put the fresh hydrogen peroxide in with the used liver, what's going to happen? So pause the video if you need to. And okay, here we go. So you can see we get basically the exact same reaction, and the speed is virtually identical. So I would also call this a speed of 4. The reason for that is that catalase, the enzyme, is completely reusable. An enzyme is not used up in a chemical reaction. An enzyme helps the reaction happen, but it doesn't get used up. So if we had a bathtub full of hydrogen peroxide and one little piece of liver, it would react the whole bathtub. It would get rid of all of it, okay? Because that enzyme is not used up in that reaction. It just helps the reaction happen and helps the reaction happen faster. Okay, that's basically the basics of the lab. So make sure you go ahead and answer the other questions in the R consensus. You can use the reading on the lab as well to help you answer the last few questions. Okay, thank you. Make sure you wash your hands after the lab.